Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. We will now go to lecture 26, where I will discuss the regulatory role of transcription factors in light induced Indole biosynthesis. What we have seen in the last class about the late steps of indole alkaloid pathway, particularly how Indole is biosynthesized from tabarsonin. So, now what we are going to see, what is the intrinsic regulation which light plays by involving different transcription factors. So, let us go to the board. So, it is only one model we will study in this class, a recent model that depicts the regulatory role. So, I just now go to the board. First, I will draw a simple diagram and then I will draw a complex diagram. So, first one, just to remind you the pathway, that means the from tryptamine and cycloganin strictocidin is formed, and from strictocidin aglycone is formed, and that ultimately leads to either azmaleucin or through the formation of O-acetyl stemadenin. I have not mentioned this compound. So, this will come in the subsequent class, where we dissect the pathway from strictocidin to tabarsonin. So, that leads to one route goes to catharanthine and other route goes to tabarsonin, and then finally, it forms Indole. So, the arrows to be added here. This is just a reminder. Now, this broken arrow means multiple step pathway, and then here what we have learned recently, that the enzyme responsible for conversion of tabarsonin to Indole are T16H, then 16 OMT, then T3O, then T3R, then NMT, then D4H, and then DAT. Where what we are going to see, that T16H, T3O, T3R, T4H, DAT, these are influenced by light. So, what happens that light and we will have to put two transcription CRPIF1 and another one is CRGATA1. So, what I am showing here is basically light induced terpenoid indole alkaloid biosynthesis in Catharanthus roseus is regulated by light. So, in other word, what we can also write that light regulated Indole biosynthesis in roseus. So, CRPIF1 phytochrome interacting factor 1. So, this basically suppresses CRGATA1. So, that is another transcription factor. So, PIF1 suppresses CRGATA1, but it is degraded by light and thus releasing CRGATA1 to activate the Indole pathway. So, that means in the dark CRPIF1 basically suppress the positive regulator CRGATA1 and that leads to no Indole formation in the dark. Okay, but when light comes, then that basically suppresses CRPIF1, that is why we are showing here an arrow, and that what happens as a result of that CRGATA1 is active, which plays important role in Indole biosynthesis. And also CRPIF1 can also directly suppress Indole biosynthesis as well. So, this is in brief how Indole biosynthesis is regulated. Now, in the next slide we are going to see in detail. So, I have to draw a box. So, or not box, we put a middle arrow. So, the left side is under the dark condition, where this represents the genes for CRGATA1 and this represents the genes for T16H2 and DAT. What happens CRPIF1? So, CRPIF1 we will put with the this is CRPIF1. So, this is binding here this is binding here, and maybe with the involvement of another yet uncharacterized transcription factor. So, what it does? It basically this is the transcription of these genes, and this is the transcription of these genes. So, it basically blocks the transcription of these genes. But when these genes activity are blocked, the basal activity is still maintained. So, the basal activity is still maintained, and that probably by the help of this transcription factor. Next, what we will see T16H2 and DAT. Okay. Transcription CR binds here, but what we see here that there is another one, that is this is basically CR GATA1, which also binds here, but in the next paragraph I will let me finish the drawing and then we will try to explain. The next one, so then what we see, the tabarsonin content tabarsonin, and this is Indole, and what we put 1, 2, 3, 4, 5, 6, 7. So, DAT, D4H, NMT, T3R, T3O, then 16 OMT, then T16 H2. So, here okay. Let me finish the drawing and then I will try to explain. So, here this side under light condition, use the same shape. Okay. This is CrGATA1. So, that unknown transcription factors binds, and this leads to transcription. And what happens here? There is a light so in under red light condition, CrPIF1 is getting degraded. Okay. And now, what we see, strong activation of CrGATA1, and then this leads to binding of CrGATA1 here, and that leads to transcription of genes for T16H2, T3O, T3R, D4H, DAT, that subsequently leads to TABERSONINE, but very strong flow for Vindolin. Okay, now one I will mark here which are influenced by the light. So, this I can mark with red. So, the diagram is complete. Now, I will try to explain this. So, this is basically a model which depicting the regulatory roles of the CRPIF1 and CRGATA1 module in light induced Vindolin biosynthesis. So, in the left side there is a, you see there is a bottom. Okay, let me use the laser pointer. In the left side, then in this side what we see that CRPF1 is likely to accumulate in the dark, because this is the dark, and it represses the expression of CRGATA1. So, it represses the expression as well as T16H2 and DAT by binding to their promoter. So, it is basically binding to their promoter site, thus not allowing the RNA polymerase to bind for starting the transcription. Repression of this CRGATA, actually this results also in the downregulation of the Vindolin pathway. Okay. So, when this is repressed, so the Vindolin pathway is downregulated, and therefore, we see that more tabarsonin is accumulated, but Vindolin is less. So, this actually the repression of this CRGA2 leads to basically the what it happens, downregulation of the Vindolin pathway genes like T16H2 DAT. So, because it is binding, it is not allowing the activity of these. Also T3O, T3R, D4H and DAT, leading to limited Vindolin production and increased tabarsonin accumulation. But one unknown transcription factors, which is denoted with this fluorescent yellow, that mediates the lower basal expression of CRGATA1 despite the CRPIF1 repression. So, that means some basal level of CRGATA1 is expressed, and that is why this is producing some amount, but it is mostly the tabarsonin, that is why I put it in big circle. Now, go to the right side where the CRPIF1 is basically degraded upon exposure of this Catharanthus rosea seedlings to light, and which is mediated by a red light sense phytochrome. So, here the phytochrome plays an important role in this CRPIF1 degradation. So, as a result of that, there will be de-repression, that means activation of CRGATA, and there this unknown transcriptor factor may plays a positive role. So, de-repression of CRGATA, this results in the activation of the Vindolin pathway. So, that means that CRGATA binds properly, and all the mRNA for these subsequent genes will be transcribed, and what happens as a result of that, Vindolin accumulates. The 7 genes involved in the conversion of tabarsonin to Vindolin are basically listed here, and where with genes which are responsive to red light, I put the red color underline, that is 316 hydroxylase, T3O oxidase, T3 reductase, D4H, DAT. These are basically the genes which are responsible to light induction. So, that is why the tabarsonin content is now less and you see the more Indole formation. So, this is basically how the light plays a regulatory role in conversion of tabarsonin to Indole, and this beautiful paper has been published by a group from Kentucky. So, I must write the reference. So, this is published in Plant Physiology. The first author is Liu et al. And this was published in 2019 in the highly respected journal Plant Physiology, volume 180, page number 1336 to 1350. An Indian senior postdoc is involved in this work, who is personally known to me. So, with this personal touch, I will end this course here. Thank you very much.